Hello, everybody. Welcome to today's video lecture, and we are covering the tissue level of organization. So let's go ahead and begin. Now, what is a tissue? Tissue is a group of similar cells that perform common function. Now, tissues are made of cells, right? And tissue make organs. So definition for organ would be, it's a structure that is made of at least two tissue types, right? So you can see, and then your whole organism is made of organs, organ systems, right? So anyway, tissue is a group of cells, similar cells with common functions. And if you look at this diagram, you can see that you will find different tissues in different organs. And because organ is made of more than one tissue type, you will find several tissues in the same organ. For example, if you take brain, you will have predominantly nervous tissue, but you will have connective tissue as well, right? So talking about organ, keep in mind, you will have predominant tissue and you will have other tissues. For example, if you are looking at a GI tract, a small or large intestine, they will be made of pretty much all of this tissue, right? They will have connective tissue and muscle tissue and epithelial that will cover lumen of GI tract. And your GI tract will have lots of neurons, right, associated with them. But anyway, so let's look at four main type of tissues that make up your body. And those four types are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Right, so again, you can see brain and spinal cord are mostly predominantly nervous tissue. You have epithelial tissue that will be found in a lining of GI tract organs, other hollow organs, and also your skin surface, epidermis. Now, muscle tissue, you will find, of course, in your skeletal muscles, in your heart, and in the hollow organs, in walls of hollow organs as well. We have three different types of muscle tissue that we will cover in this chapter. Connective tissue, the example would be your fatty padding tissue, your bone, your tendons, even blood is a type of connective tissue. Now, during embryonic development, the cells will differentiate into three germ layers first: ectoderm, mesoderm, and endoderm. And then all your organs and organ systems will be developed from these three germ layers. And there is a lot of, kind of different structures and organs that develops from those layers, but let's just look at major ones. For example, ectoderm. Now, from ectoderm, you will develop your epidermis, your skin cells, and interesting nervous system as well, right? So ectoderm, epidermis, and nervous system. Mesoderm will give rise to your muscles, like cardiac, skeletal, smooth muscles, and lots of connective tissue, right? For example, red blood cells over here, bone, cartilage, right? And some epithelial, well, we will not call it epithelial, but endothelium. So this is epithelium, develops from ectoderm. Endothelium will develop from mesoderm, and endothelium will line your blood vessels, and right, so and form maybe some membranes, serous membrane, synovial membrane. No, not synovial, serous membrane. And endoderm endoderm will form your digestive system and your respiratory system, and also some glands, like digestive glands, endocrine glands, adrenal glands. So again, as I said, it's a lot to memorize over here, but the major organs for ectoderm is epidermis, nervous tissue, mesoderm, muscles, connective tissue, endoderm, respiratory, digestive tract, right? Now, before talking in more detail about those tissues, let's see what membranes are. Actually, membrane is a layer of cells that the function is to cover something, right? It can cover your entire body, and then we call it cutaneous membrane, or it can cover your open body cavities, will be mucous membrane, or it can cover your closed body cavities, and this will be a serous membrane. And another membrane in our body is synovial membrane. Now, mucous, serous, and cutaneous membrane are made from connective and epithelial tissue, and they consider to be really like simple organs. Like your cutaneous membrane, your skin is your organ. So remember, organ is a structure that is made of two different tissue types. Synovial membrane is made of connective tissue, right? So connective tissue membrane is synovial membrane, and epithelial connective tissue membrane is mucous, serous, and cutaneous. Okay, so we're gonna start with epithelial tissue. The general structure include highly cellular. So you can see cells are touching each other. So we have many cells. And cell form cell junctions. There is a polarity. That means on a tissue and on a cell as well, we will have basal layer and apical layer. So here's the tissue from this point where this darker line is up to the top, and this is the basal cells, and on the top you will have apical cells. So that's a polarity of tissue. And also if you look at individual cells also will have different poles, apical and basal pole. And those might have slightly different structure and function. It's avascular. That means we do not have blood vessels. Also rapid reproduction. General function of epithelial tissue is defense, transfer of materials, and secretion. So we just mentioned that there are junctions between epithelial cells. We have three major types of junction: tight junctions, then anchoring junctions, and gap junctions. Tight junctions separate cells into apical and basal compartments and block movement of substances. The cell membranes, they're pretty much fused together, and it does not allow substances to move between cells. So that make this membrane very efficient. So if you see this like a little box here represent a cell, and here we have another cell next to it, and these little squares represent tight junctions. So those are between cells. Anchoring junction include desmosome, hemidesmosome, adherents, and so you can see the pictures over here. So those you will have transmembrane proteins between cells, right? So that will allow you to stretch cells a little bit, right? Give it a little bit of give between the cells. And gap junction shown over here allow passage between cells, right? This anchoring junction like desmosomes, for example, they like like a zipper in between cells, right? So you can really stretch, pull cells away from each other just a little bit, just to prevent the tears within your epithelial membrane, for example, in your skin. Now, how do we classify epithelial tissue? It can be classified based on the shape of the cells and a number of layers, I would say. So if you look at this diagram here, epithelial tissue can be simple when we have one layer of cells. So here, this one is simple, this one is simple, and this one on the bottom is simple too, because we only have one layer of cells. Now, this line, kind of like a little bit purple line, this is called basement membrane. So all epithelial cells, they are resting on a basement membrane. And opposite to basement membrane will be free surface. So we call this area free surface. So you have basement membrane and free surface. And this also tell us, we have polarity in those cells, two different poles, basal pole and apical pole. But the second way to classify is based on the shape of cells. Now, those cells are flat. Flat in anatomy means squamous. So it's a simple squamous epithelium. Now, this epithelium is also squamous, but it has several layers. So it's stratified, right? So this is stratified squamous. This one, cells are cuboidal, shape of cube. So this is simple cuboidal. This one, we have two layers. So it's stratified cuboidal epithelium. Those cells look like columns. So this simple columnar epithelium, and this one is stratified columnar epithelium. Now I want you, so just to recap, epithelial tissue has at least two words in its name. First word need to be either simple or stratified, right? And the second word is a shape of cells. So simple squamous, simple squamous, stratified squamous, simple cuboidal, stratified cuboidal, simple columnar, stratified columnar. I also want you to look at this picture and see those basal cells. Sometimes they might have a different shape. For example, this can be cuboidal over here, or even maybe look like a columnar. But when we classify epithelial tissue, we're only looking at apical surface. So because on top we have flat cells, it will be stratified squamous. Doesn't matter what shape of my cells are at the basal end. There is another type of epithelium, it's called pseudo stratified columnar. So this one is not simple, it's not stratified, but when you look at this slide, and if you have a microscope and you look at the slide, histology slide, it will look like you have 2 or 3 or even 4 layers of cells, but that's because cells are different heights. So some cells are tall and some cells are short, and nucleus will be located on the different levels. So let's see this nucleus on the top, this nucleus on the bottom, and it looks like we have 2 layers of cells, but every single cell touching basement membrane. Like over here, look, this second layer, the cells do not touch our basement membrane, but here every cell touching basement membrane. That's why we call it pseudo-stratified columnar, right? Okay. Now, what do we need to know about tissues? We need to know description, and that means structure. We need to know function and common location. So a simple squamous epithelial, a single layer of flat cells, right, because it's simple, with disc-shaped central nuclei and sparse cytoplasm, the simplest of the epithelia. So if you see those lines, right, this line over here, those are cells. Now, what is inside? This is not the tissue that we need. Those are actually air spaces. So there is air inside, but the sac itself, you see, like a wall of a sac is made of simple squamous. And those darker dots are nuclei. So each cell will be surrounding this nucleus. So we have a cell shown kind of in a pink color, right? Look, just like a line, really. And then a little darker dot inside is nucleus, right? And cells are connected and they make those alveolar cells or air sacs. Now, because this, and of course it's on a basement membrane as any epithelial cells. Now, because it's so thin, the major function will be diffusion or filtration. So something that allow substances to go across the cells easily because we don't have many layers, right? Diffusion and filtration in sites where protection is not important because you cannot protect much with one layer of very thin flat cells. Secretes lubricating substances in a serous membrane. And I'm reminding you, serous membrane will cover your thoracic cavity and abdominal pelvic cavity. Now, location will be where we need this diffusion and filtration. So kidney, for example, we need filtration and we need diffusion too. Air sacs, we need diffusion. Lining of the heart, blood vessels, and lymphatic vessels, lining of ventral body cavities, so they form a serous membrane, right? Now, please pay attention that the structure and function, right, they are inseparable. It's called the principle of complementarity. Principle of complementarity tells you that if we pretty much know structure, we can predict the function, or if we know function, we can hypothesize structure. So they are connected. All right, you cannot have like 150 layers of cells and assume that it's gonna do diffusion or filtration. It just does not fit, the structure does not fit its job. And if you know structure and you know function, well, then you can pretty much predict where it can be located, right? Where do I need diffusion? Where do I need filtration, right? So pay attention to this, three criteria that we cover: description, function, and location of each tissue. Next tissue is simple cuboidal. So it's simple, so it's a single layer of cube-like cells with large spherical central nuclei. So again, this purple represent basement membrane and then cuboidal cells, and those circles inside are nuclei. Now, if you look at this tissue over here, we have tubes again. So you see, this is one tube, here's another tube, here's another tube, and all those tubes, they are made of cuboidal cells. Within this cuboidal cell, you will see nice round nucleus. Now you see this cell over here, it seems like no nucleus. Well, there is a nucleus. It's just how slide is made. Maybe it was made in a part of the cell that, nucleus may be on another pole of the cell. Right? So sometimes you cannot see nucleus, but every cell has it, right? So those darker parts are nuclei, nucleus. Now here's another tube. And again, it's made of cells. Each one has nice round nucleus. In between cells, we will have connective tissue. We might have some other cells, right? We might, but what we interested in into these cuboidal cells that make those tubules, and this is photomicrograph of kidney tubules. Now, what would be function? Function would be secretion and absorption. So if you look over here, diffusion and filtration, well, now cells getting larger, and they do not feed a function of secretion, or I'm sorry, a function of diffusion or filtration. But secretion is pretty good, right? So you can make some product, you can secrete it. And actually, many glands are made of cuboidal cells. So where we have secretion, where we have absorption. So we will find it in kidneys, all right, because we will need reabsorb stuff in the kidney, for example, and we will secrete something in kidney. And also glands, as I told you, right? Secretory portion of smaller glands. Also, ovary surface is covered by simple cuboidal. So over here you see kidneys, and they show you they have those simple cuboidal epithelium. Simple columnar. So we would have one layer of cells. Cells are column shape, and it will have round or oval nuclei. Some of this epithelium will have extensions on it, apical surface. It can be either cilia or microvilli, right? Many of them will have mucus secreting unicellular glands or goblet cells. So here's a histology slide. You can see tall cells with nice round nucleus over here, right? That's a basement membrane shown in a darker color, and that this will be free surface. So function, absorption, secretion, the secretion of mucus, enzymes, and other substances. Ciliated type propels mucus or reproductive cells by ciliary action. So the function, absorption, and secretion is the same as cuboidal, secretion and absorptions. But we, because we can have, for example, cilia for movement, then we will find it in the parts of your body where you need a movement of, mucus across the surface, for example. So this is very common epithelium in a digestive tract, and then it will be non-ciliated. It will be either simple columnar, or it can be simple columnar with microvilli, right? So it's from a stomach to anal canal. You will find it in a gallbladder, excretory ducts of some glands. Ciliated will be found in a small bronchi, so somewhere where you need the cilia to move mucus, right, in the bronchi, uterine tubes, because you need to move actually ovum, and some region of the uterus. Next epithelium is pseudo-stratified columnar. Now, if you look at the histology slide, you see, well, it does look like it has 1, 2, 3, maybe 4 layers. And it's hard to see that every single cell touching the membrane, but you just need kind of to be familiar with this type of epithelium and be able to identify it. And plus, stratified columnar is very rare. So if you see that looks like many layers and you see big, tall columnar cells, that most likely gonna be pseudo-stratified. So it's single layer of cells of different heights, some not reaching the free surface, but all of them touching basement membrane. Nuclei seen at different levels may contain mucus secreting cells and bear cilia. So this cell over here is a goblet cell. This is the mucus secreting cells, and on the surface you see cilia. Now function, secretion, particularly of mucus, propulsion of mucus by ciliary action, and location, non-ciliated type in males, sperm carrying ducts and ducts of large glands, and ciliated variety lines the trachea, most of the upper respiratory tract. So very often we call this epithelium respiratory epithelium because it's found in the most of your upper respiratory tract. And then in a small bronchi, I went back, in a small bronchi, you will have the simple columnar. Also will be ciliated, right? So we go in your, let's say, your respiratory tract. If you start with nasal cavity, so in your nose, in a nasal cavity, you will have pseudo stratified, right? Then from nasal cavity, you go to pharynx, and pharynx you share with your digestive system. So you cannot have this epithelium because your food will damage it. So you will have different stratified squamous. And then when you get to a trachea, right? Larynx, trachea, main bronchi, secondary bronchi, even tertiary bronchi, you will have pseudostratified. And then when you get to smaller bronchi and bronchioles, then you will have simple columnar. And then when you move to alveoli, way kind of, down to your lungs, where gas exchange happen, you will have simple squamous, right? Okay. Next epithelium is stratified squamous epithelium. Now we are in the first stratified epithelium because everything that we looked at before, let's go back. So all this epithelial tissue that we look was, so we started with this, was simple squamous, right, simple cuboidal, simple columnar, and pseudostratified is actually simple, right, because it's one layer, it just cells of a different height. And this is the first stratified epithelium. There is a pretty common epithelium, right? That is our, just a second, I just want to see. Okay, stratified squamous, right? So let's go ahead and see what we have. So we have basement membrane, right? And then we have many layers of cells, and close to basement membrane, we might see cells that look like cuboidal, right? Those look like cuboidal for me. And then when you're getting to the top, and they became more flat. And remember, we're only looking at the apical surface. If those cells on the top are flat or squamous, then it's stratified squamous. So basal cells are cuboidal or columnar, metabolically active. Surface are flattened, squamous. In a keratinized type, they are dead. And if it's non-keratinized, they will be living cells on the surface. Basal cells over here on the first layer of this epithelium, they divide by mitosis and produce the cells of the more superficial layers. Now, if you see so many layers of cells, you can predict the function. So it, would be secretion a good function for this epithelium? No, right? Because look, substances need to go through so many cells to be secreted. Lots of energy need to be used, and your body doesn't want to use extra energy if it's not needed. So secretion is not good function. Absorption. Well, for example, you have some molecules here, I need to absorb them through this many layers, and also probably not a good function. Filtration, again, it's too many cells. Now, what would be a good function for this epithelium? Protection. When you have so many layers of cells, that's good to protect your body and your organs. So it protects underlying tissue in areas subject to abrasion. Now, location, you will find where you need this protection. Non-keratinized will form lining of esophagus, right? Esophagus is your it's a part of your digestive system, right? The food goes from esophagus to the stomach. You need protection. Why do you need protection? Because your food might be, if you eat solid food, and of course you chew it, you mix it with your saliva, but still it can damage your esophagus and pharynx. So esophagus, mouth, vagina, that will be non-keratinized. Keratinized forms epidermis of the skin, that is your dry membrane or cutaneous membrane. And this is what a micrograph shows you. This pink portion, the whole portion over here is stratified squamous, and that's connective tissue underneath. And those dark spots are nuclei. So cells are attached to each other. They have all these, tight junctions, desmosomes, gap junctions. So cells are attached. So if you see dark oval structure here and dark oval structure here, those are not cells. Those are nuclei, and cells are, cytoplasm and cell membrane surrounding it, and there is connection between cells. This epithelium is called transitional epithelium. Now, this transitional epithelium was not a part of the diagram over here that we covered, but it would be in it's a stratified, right? So we would put it in a division of stratified epithelium. And it's resemble both stratified squamous and stratified cuboidal. And basal cells, cuboidal or columnar, surface cells, they have like a dome shape, and they also squamous-like, depending on the degree of organ stretch. So this is how we would draw a diagram. On the top, we would have cells that really like cuboidal over here. Some might be squamous, but in the squamous cells, we don't have this nice round nucleus. So really, apical surface look like cuboidal, and we have many layers. Now, a true stratified cuboidal has only 2 layers. Here we have many layers, and it looks like cuboidal on the top. Then it's transitional. Transitional epithelium can stretch. So you would, so the function. So let's look at location first. It lines the ureters. That's a ureter. A urinary bladder, and a part of the male urethra. So now what you need for this organ to be able to do, you need them to stretch, because when your urinary bladder is empty, right, then this epithelium will look like those upper cells will look like cuboidal. But when you have urine in a urinary bladder and the walls stretch, this top portion will look like squamous, right? So this is a rare epithelium, and it's only pretty much found in a ureters and urinary bladder, and only in male urethra, just a part of male urethra. So stretches rapidly and permits distension of urinary organ by containing urine, right? So that the function is to be able, it has ability to stretch, right? Okay, so now here we have pretty much the summary of the epithelial tissue. Simple squamous, simple cuboidal, simple columnar, pseudo-stratified columnar, stratified squamous, stratified cuboidal, stratified columnar that we didn't cover. So we will talk about it right now, just very briefly, but this is our transitional epithelium, and lines the bladder, urethra, and the ureters. Now, why we didn't cover stratified cuboidal and stratified columnar? Because it's a very rare epithelium. And a stratified here and here, we really have only 2 layers. Only 2 layers of cells. So here in stratified squamous, we might have 100, 150 layers. We have many layers in the transitional. Stratified cuboidal and columnar, we have 2 layers. And really what it does, it makes stratified cuboidal make bigger glands, and stratified columnar found also in the glands and male urethra. So rare epithelium. And it's easy, secretes and protects, right? So now another type of epithelial tissue, because we were talking about just epithelial tissue in general, but mostly it was covering epithelium. However, epithelium also made glands. So glands are made of epithelial tissue. So, and that epithelium is called glandular epithelium, made of cells modified to make a product and secrete product. When we're talking about glands, we have endocrine glands and exocrine. Now, endocrine glands secrete hormones in your bloodstream and lymph. Exocrine glands secrete their secretion, their product, through ducts. So we must have a duct, and then we call it exocrine gland. And secretion moved through the duct to the surface of your body or inside the lumen of hollow organs. And endocrine glands, again, they do not have any ducts and they secrete hormones in bloodstream. Now, glandular structure, so the gland itself can be unicellular, very rare, like a goblet cells. And when this whole gland is made of one cell, or multicellular, many cells. And now when we look at different type of glands, we classify them based on a structure of gland cells and a structure of a tube. So here we see all glands with a simple duct or tube, simple duct, or it can be compound duct. So if a duct is simple, it will be always simple gland, simple, right? So it can be simple alveolar. Now the gland cells, they look like a sac, right? Or simple branched alveolar. So you see those sacs, those are alveolar, or it can be simple tubular, or simple branch tubular, simple coiled tubular. So the first word in the name will be simple if we only have kind of one duct over here. Now those cells that show like lighter color, they do not secrete. Those that show in a pinkish color, those are secretory cells. Now, if we have a duct that is branching duct, it will be compound. So this one, compound alveolar. Another name for alveolar is acinar. So you see, this is the duct. This like three ducts come together to make one single duct, right? So it's compound. And a secretory unit is alveolar. This is compound because, again, we have three different ducts making one, but this has alveolar and tubular secretory unit. So it's a compound tubular alveolar, or this one is compound tubular, for example, right? So glandular epithelium makes glands, including the secretory units and their duct. So you can see this is pretty much what simple cuboidal, right? Simple cuboidal, simple cuboidal. If we have a large duct of the gland, they might be simple columnar as well. Now, method of secretion. So we talk about endocrine versus exocrine, right? Now, your exocrine glands can secrete through merocrine secretion, apocrine, or holocrine. Now, merocrine is the most common, shown here on the top. When cell synthesize product, right? It's synthesized on the Raphe and then move to Golgi apparatus, and then the product moves towards the surface of the cell within secretory vesicles. And it's released through exocytosis, right? So we form vesicles, release through exocytosis, then it will be merocrine secretion. And example would be sweat glands and specifically eccrine sweat glands. So this is the one that doesn't produce body odor. So those are glands that their function is to cool down your body. So when you're hot, they secrete sweat and it cools you down. Another secretion is apocrine secretion. And if we give example, it would be sweat glands, but those that found in axillary and genital regions, and they produce body odor. Apocrine glands are also mammary glands that, produce milk when we breastfeed our babies. Now, over here, you can see the secretion accumulates near apical portion of the cell, and then it's pinched off with the part of cytoplasm. So here cells make their product, again in the rougher, move to Golgi, and then you do not really form vesicles, but you accumulate the secretion at the apical surface and it's pinched off with a part of the cell. And holocrine secretion, when we have destruction of entire cells. So entire cells are gone, right, when we secrete product. An example would be sebaceous oil glands, right, it's your oil gland. So here are holocrine secretions. So you make product, right, and it moves towards the apical surface, but now the whole cell is destroyed and you need to replace this cell, right, pretty much. That's oil glands. Those are methods of secretion. Okay, so we covered epithelial tissue, right, of this chapter. And this is where I'm gonna stop this video. And I will record the second one where we will talk about connective tissue, muscle, and nervous tissue, right? So thank you for watching, and I hope it was helpful.